Okay, so today I'm going to talk about DolphinNext. This is a graphical user interface for reproducible data analysis. So last couple of weeks we were talking about uh, preparing the files and mapping them to genome and do quantification on those files and do some differential expression analysis. So when you look at RNA-seq data, so you have these kind of steps like pre-processing of the data. Usually your data are ready to align, but sometimes when you get your data, what we do first is quality checks. So we're just using like a fastqc command to prepare some quality files. And by looking at those files, we can understand if there is something wrong uh, in our reads. If there's something wrong, you can send them back to sequencing facility to resequence, for example. Or you get them and you realize that there are some problems in 5' ends of your reads. So what you need to do is you need to trim 5' end, or sometimes 3' ends are not good. Or sometimes if it is a smaller RNA-seq, you do adapter removal. Or if your fragment sizes are small, even it's a bulk RNA-seq, total RNA-seq, you might need to remove those adapter sequences. Or sometimes some of the qualities are bad. You want to remove them before alignment to reduce some alignment errors, mapping errors. So these things are not only for RNA-seq. When we were, could you mute, please? Okay. So when we are preparing our pipelines, we realize that most of those steps here in the pre-processing step or processing step or any of them are similar in many different pipelines. So, and then we realize that this is the case, then what we need to do is we can reuse all the steps in other pipelines. So for example, we created RNA-seq pipeline I'm going to show you today, but we created ChIP-seq pipeline too, or some ATAC-seq pipelines. Then in those pipelines, we realize that those steps are all the same. So that's one of the reasons actually we created DolphinNext is reuse our previously defined or previously created steps in other pipelines. Here in the processing steps, we split these all cycle data analysis and processing step with these boxes here. But first, pre-processing is just to prepare your files for processing. Means in pre-processing step, you're just removing, for example, adapter sequences, barcode sequences, or if there are some unique molecular identifiers in your reads, you actually detect them and put them, group them, and put the UMI into the header, and then you're just removing this. You just collapse all the reads using UMIs. So, and after you prepare all these reads, then you are ready to align them to transcriptome if it is RNA-seq, for example, or you can align them to genome if it is a DNA or chip. Or sometimes I had a question about like, okay, which one is better? Like, should I map them to transcriptome or genome? Because you're teaching both. So there is something here that you have to understand. So there is no better or worse here. So when you have your reads, so you need RNA-seq reads, what you need to do, for example, if you want to quantify mRNAs there, you need a annotation. So, but those annotations might be limited. So somebody made those analysis, so they curated those annotations, and you have the latest annotation, but that annotation might not be complete. So that's why what I tell people usually is this. So first, okay, use the best annotation you can find and align them, quantify them, align them to transcriptome using RSAM or other programs, and align them to transcriptome, quantify them, and get your tables. So this is one thing. But to check if it is complete or not, you need to align them to whole genome with a splice junction support aligner like HiSet or STAR. So, or you can do it TopHat, but we don't use TopHat anymore because it's extremely slow. And STAR is, the quality of STAR is a little bit better. So you can use star or high set according to your purpose. But here, so what I'm saying is when you map them to all genome, you can go to IGV and go sites manual and look if there's something interesting. And you can use other tools like cufflinks or transcriptome assembly softwares like StringTie. That StringTie is going to use your own reads to assemble the transcriptome. So you can use that transcriptome to quantify again. So that transcriptome will come from your own reads. So, but the problem there, it can be extremely noisy. It's not going to be curated. There'll be lots of like transcript that they are not even exist. So there can be like short transcripts. But the reason that you are doing this, if you are curious about like some sites that are not in your annotated transcript that you found somewhere else, so you can do this kind of transcriptome assembly, then quantify them, find regions that they don't exist in existing assemblies, existing transcriptomes. Then you can see maybe something different is going on in your own reads, in your own experiments. So that's why, but this is kind of rare. But if you're doing something rare, if you're doing something new, if you are applying a new drug, or if you think that what you're doing is totally different than what is done before, then you might also consider doing this. So this is something that you have to understand about why we are using transcriptome assembly. And after you do these steps, data processing, so you get the tables. And there are also other steps here, like after you get your tables, you can, before doing this in processing, we had the quality checks about just the reads before you process your data. But after processing, you also need other like post-processing quality checks. You need to do that too. Like for example, you mapped, your reads were perfect, they aligned somewhere, but then when you do the quality checks, it was a bulk RNA-seq, but you get only 10% of your transcripts covered in your RNA-seq data. So this means that maybe the amount of the RNA wasn't enough, or there might be other problems. Even sequences was perfect, even the fragment sizes were perfect, but there was something bad in the biology or experiment. Something was wrong, and then you get like wrong quantifications. So that's why you need to do pre-processing quality checks and post-processing quality checks. If everything is like uniformly distributed or like distributed well in all of your transcript in bulk RNA-seq, you're good. But if you're doing, for example, single-cell RNA-seq, according to the chemistry of your single-cell RNA-seq data, you can go up to 30, you can all cover up to 30% of your transcripts. So you have to think about that too. After in the quality check step, you have to see how many transcripts you could cover with your single-cell RNA-seq. For example, if you want to cover most of the RNAs in your cell type, then maybe single-cell RNA-seq is not good for you. So you have to also think about that. And there are other checks. For example, if you did the transcriptome assembly, and there are other programs to do quality checks on that assembled transcriptome. So it's checking the size of the transcripts, checking the average length of the transcripts and the number of exons, those kind of things. And for example, if you have the number of exons are all skewed to 1, means it's not, transcripts didn't assembled well. So those kind of things that you have to do after processing the data. And in the downstream analysis, it's a world there. So after, like we are doing, for example, pre-processing, processing, post-processing, and we in DolphinNext, we entered downstream analysis a little bit more, that we do differential expression analysis or those kind of analysis in DolphinNext too. But there are many other things that you could do yourself, or there are many third-party tools you can use, like Jupyter, for example. Use Jupyter, upload your file there, and use other people's like analysis methods and get your results. But again, here we are trying to automatize everything as much as we can to reduce all these pre-processing, processing, and post-processing step workload in these steps, and then give you more time on downstream analysis. Even you're a bioinformatician, this will help you a lot. Like doing these kind of things automatically will help you a lot. And if you have a problem, you can go back and fix a problem, rerun them again. So this is like a cycle. So when you have a large projects, those large projects have like a couple of needs. They require capturing sample metadata. And in DolphinNext, we created a new thing called DMeta, Dolphin Meta, that I'm going to talk. So you can actually keep all this metadata and attach to your samples. And this metadata is very important because sometimes you collect data from one location or apply different like kits while you're designing your experiments or doing sequencing. You need to correct batches if you did this in different batches, or you are working with another lab, they did similar experiments, and then you want to combine the data coming from different geographical locations, then you need to correct the batches to be able to do some analysis on them. And if there are problems, you need to troubleshoot with using this metadata. For example, they are saying that this data is blister coming from vitiligo patients, but you are checking they are closer to healthy patients, for example. Maybe there was a mislabeling in those samples. So you need to troubleshoot by keeping this metadata. And the pipelines that you need to create in the future, we're going to give an example. DolphinNext will be an example for you. In the future, you're going to see many different pipelines that you might want to use, like created for with SnakeMake, or like different programs like Nextflow that we are going to show. And but when you're choosing your pipelines, they need to be highly parallel when you are doing a large project. Even you have like more than 10 samples, you have to do this with highly parallel support. And the pipeline should be self-monitored. For example, when you have 100 samples, you're aligning them, and when you get an error in one of them, so the pipeline has to check the error and then resubmit that job again, because sometimes the hardware you're using might have other problems that you cannot control. But when you resubmit the job to the cluster, it might finish successfully. So that's why if you have a pipeline system that is monitoring itself, if there's a problem, it can resubmit your jobs, or there is a problem in your file, for example, that there's a corruption there, and the pipeline is going to throw an error to you. And after you see the error, you can put the original file back there. And then when you resume your pipeline, it will finish it. So these kind of functionalities are very important in a pipeline system. If you don't have it, you can lose a lot of time. Sorry, is there a question? Okay. So I'm going to talk about versions. Versioning is very important in a minute, and handling large number of samples to finish your analysis or data processing fast. So the problem here about like what we have that we realized when we started doing this kind of analysis for different labs in time, we realized that all these things are changing in time. Like, for example, the operating system you are using while you are doing your analysis changes. And somebody, for example, published the methods 6 years ago, 5 years ago. You published your own method yourself. And if you want to reproduce your own results, if you don't have the same operating system, for example, the versions of the programs that you installed on that operating system will not be the same. Or the language that the people used has a version, for example, and that language, that version, for example, C compiler they use while doing that, while writing that paper, but in your operating system, that version doesn't exist and you cannot compile that code anymore. Means you cannot use their program. Or The things in time changes. So to be able to go back and reproduce your own results or someone else's results, you need to have a snapshot of everything you have here somewhere, and then you should actually use that exact machine on top of another machine on your own machine to reproduce those results. And I'm going to talk what I mean. So even those people or yourself created an open source code, it doesn't mean that that open source will work in the future. Even you share this open source on GitHub, this is GitHub, even that source, that GitHub means that the people are sharing their own code to the world in this repository system. Actually, it was an open source itself before, and Microsoft bought it and now, and they are maintaining it. So even it's shared there, it doesn't mean that it will work forever. That I'm going to tell you how you can actually create pipelines for your own method to make it available forever. And Travis is that we are always using, the people started using this to test your own code on different computing environments. So this helps us a lot when we are developing our own method. When we change anything in our method, Travis automatically tests our methods, for example, and then it tells if there's an error, we fix the error and then submit. So this is increasing our efficiency. There are all the things that I'm talking about here are increasing our efficiency. So then to get the snapshot, all this machine, a couple of years ago, new methods, new software came, new platforms came. They are called Docker and Singularity. These are like very lightweight virtual machines. Virtual means those are not real hardware, but they are installed on top of a server, but they have their own operating system, whatever you choose. So it means when you publish a method, if you install your operating system on this, they are called containers, on these containers, and you're going to have the snapshot of the machine. And then this is just one text file. When you have that text file, it's like couple kilobytes or couple bytes, a couple hundred bytes file, like very small files. So when you have that file, you have the power. It means you can actually have that machine forever. You can install everywhere. So you have the actual recipe of that operating system and machine, and all the language, all the versions, all the metadata, all the parameters you can actually deploy inside of a container to reproduce what you did before. So, and Nextflow is using the Singularity and Docker to create pipelines. This is also, you can run Nextflow on command line, like you did other software you are using, RSAM or Star, but you can actually create your own pipeline in a file, like if you know coding, and then you can actually share that pipeline with the people with your paper in your method. The people actually start from the raw files and process the data and then get the table, for example, at the end. So you can actually create these kind of pipelines with Nextflow. And after you process the data, what we are usually using to analyze the data are Studio or Jupyter. So these are helping us a lot because science improves every day. And once you process data, you can always improve how to analyze your data with new algorithms, new methods, and share these analysis results in your, again, repository in GitHub or Jupyter notebooks. So here, like, I just want to give an example that I wanted to reproduce this paper, and this was published a couple of years ago. It's not that old, 2016, so or something like that. So here it's a single cell RNA-seq data. They have beautiful data. Everything is beautiful, but the software version they used at time were here. Versions are here. So, and even I installed many of these versions, some of them are not even installable today, are version 3 or something. So, but they published everything they had about how they did analysis from 0 to the end. So But the thing is, it's almost impossible to rerun what they did in the GitHub. So it took 1 month for me to run what they did. Even like I couldn't install everything here because some of them are not supported anymore. And what I did was I had to rewrite with all these things with new Surat. Surat changed a lot too in time. This is a single cell RNA-seq analysis package in R. So there are other things here change a lot that almost it's almost impossible to reproduce exactly the same results. But I get like a similar PCA plots here. But again, it was like even you have a good mindset to share everything you did, but in the future, in a couple years, actually, it's like less now because these tools are improving a lot. You cannot reproduce these results very quickly. So it took 1 month for me. And then what I did, like the reason is that because they are like putting, when they're writing their code, they are not that thoughtful about other people are going to run them in the future. For example, they put their own uh, folders into their code that they run in their own environment. These are not going to work on me. If you're not a programmer, for example, if you just want to run their code, it's impossible. So you have to change lots of things in their code. This is just an example, but even the versions are not going to work for you. So you're just gonna this is useless. So what I can say, even these people are great people, but whatever they deposit here is almost useless. You have to redo everything yourself. So when you try to install, you get these kind of lots of errors that it's not installable. So to I want you to convince today that in the future, when you become a PI or if you go to the industry, you're going to realize that in time, things change. But sometimes you want to reproduce or rerun what you did before, exactly like how you did. I'm going to give another example. So I was working on a bear project and they gave me a group of bear samples. So they were like hibernating bears, and they collected samples when they're awake, and then we compared like 3 years ago. And I wrote my pipeline here in DolphinNext, and then run the pipeline and then get the results, and then gave them back. They loved it. So we even applied for a patent using the results. But then they realized that, hey, we need to do this thing again with different tissue. And we need to collect samples from kidney or from liver. So they sent new people to the mountain and they collected some other samples and they brought them back and they did that. Can you do exactly the same analysis? And I really forgot that I did this on DolphinNext because DolphinNext were like new, brand new that time. I thought that this was before DolphinNext, but I realized that this was my first pipeline that I created in DolphinNext. And what I did, I saw that and I just changed the files, rerun the pipeline and get the results like without doing anything. And then I just share the results. So I just changed the file names, all the parameters, all the software versions, everything from 3 years ago were already there. So this made my job extremely easy, and I didn't even need to think like too much about that. I just changed the file. So this is lifesaver when you start using these kind of tools. I'm not talking about only DolphinNext, but I want you to realize that you need a way of organizing your pipelines like this that anybody and yourself should be able to reproduce what you did before. So because you're going to realize, maybe you already did, that this is a like research data has a life cycle from creating and processing the data, analyzing it, preserving, sharing with other people, and reusing your own data. And with the newly generated data that you want, you always, or you most of the time, you want to compare your new data with your old data if everything is correct. Or so this is a life cycle. Sometimes you create data, process, new patients come, for example, you create again, you process, analyze data. So this is ongoing project, especially when you're a PI. I know people are using data from like 10 years ago that they are, even Arthur found, for example, some microarray data and comparing with RNA data. So those data published like 15 years ago or so. So sometimes you need this kind of method to understand what you're working on in a long time period, like 15 years, 20 years in the future. You're going to see that these kind of tools are giving you that power to reuse your old data. So for every result, you need to keep track how your data is produced. So even you are the most needy greedy person who keep tracks of everything in the documents, you always skip something. Like you change a parameter and then rerun things, and then you forget put that parameter change. So these kind of things can affect your success. And we bioinformaticians always do some manual manipulation steps to do things faster. Like, for example, there's a file here, a table, and you just remove one column from the table and create another table. And when you do this once, it's okay. But when you do this thing, because they're asking you new thing, hey, can you map this thing to this new annotation? And you do this, and then that you come to that manual operation, and then you have to go there and do it again. So when you do this 100 times, it's just reducing your efficiency too much that you don't want to go and do it anymore. So, but when you avoid manual data manipulation steps, and when you do these things in DolphinNext, then everything will be automatic. You're just going to change the files, and then DolphinNext will do the job for you. And archiving the exact versions of all programs you're using, especially external programs, or also internal programs, that, for example, when you do differential expression analysis every other week, they are changing something in DE-seq2, for example. They have a minor version. When you take two minor versions, sometimes you don't get the same results using exactly the same version, but the minor versions are different. You cannot reproduce your results. So this is extremely important. You have to get a snapshot of all the versions you have. Even you note them, sometimes you cannot even install right. Those are becoming very cumbersome, because when you install one program, another program like gives an error that, okay, hey, this program is not working with that program anymore, or something like that. So it's crazy. So that's why when you do something, it means that it worked, that you have to get a snapshot of what it worked, and then it will work forever. So think about that. And version controlling is also important about the scripts that you are writing, because they also improve. You're a scientist. Every day you think a new thing. When you are walking, for example, and then you apply that new thing into your script, and then you should version them, because sometimes you want to go back. And if you don't do this kind of versioning for every changes you do, then you cannot actually reproduce you did a week ago, or even a week ago. So, and providing an access to other people that they can actually utilize what you did, your pipelines or your data, with fair standards I'm going to talk about in a minute. Okay, so to be able to do everything, we created DolphinNext, and it's an easy to use web interface to create pipelines with a drag and drop interface, and deploy your code to GitHub to share with world in your, together with your publication, and also execute those pipelines anywhere you want in the world. So this is a very flexible and easy to use application that in the future, when you go somewhere else, you can install DolphinNext into your own laptop and use it, or you can still use our UMass version, or you can tell your IT people in the future that they can install it for you, that all your university might use it. So there are now like 2 companies are using DolphinNext in their own system, and they install them in themselves, and 2 more companies are, like, we are still talking, actually they are going to use, and there are many universities actually started using in their own institutes. So the DolphinNext supports all skill levels. You could be anything, like you can be a statistician or computer scientist, or computational biologist or any field you are in, any skill set you have, you can find something to do here. For example, if you're a bioinformatician, you can create your own pipelines, share with your collaborators or people you're working with. Or if you are a bench scientist, you can use existing pipelines and also create your own pipelines easily by like choosing existing steps, is existing processes, and connect them and create your own pipeline, version them, give a name, and then use it yourself. Or you can just use our RNA-seq pipeline, just press run button, fill the form, and submit the job into our cluster and get your reports. So it's up to you how you want to use. But I know people that I started getting like emails from people on LinkedIn that they find a bioinformatics job before when they were in UMass, they were a bench scientist, for example. After they took our boot camp, they became a bioinformatician. I'm like seeing them and feeling extremely happy. So DolphinNext is working on top of Nextflow. Nextflow is a pipeline manager that supports cluster systems, any servers, cloud like Amazon or Google. So we are actually leverage from Nextflow to utilize all the services that they support, many services they support, and give a user interface to the people to make things easier, and we abstract that level. And because this is kind of hard, there's a huge learning curve to write in Nextflow. But with DolphinNext learning, there is still learning curve, but a couple hours, you can actually understand what's going on here and run your own pipelines yourself. Today, actually, hopefully, you will learn how to create your own pipelines yourself using DolphinNext. Excuse me. So, and we realize that things are missing in DolphinNext. Means we have big projects, and in those big projects, they procure the samples and do the library preparation. Then scientists are doing this library preparation and sequencing and checking bioanalyzer data and everything. But this is also a workflow here going on. And we wanted to keep track what's going on in every step about a sample, when sample is collected to the analysis. And we want to attach every single information to that sample that the different people in different working in different part of the organization can actually update some status of the sample. For example, if this person is working in library preparation in the bench, that when that person gets the data and prepares the library and run the things on bioanalyzer and get the results, send these results back to the Dolphin the meta program, and then the other people who are working on the same samples can actually see the status of the samples. If the bioanalyzer results are good, everybody can actually access it information and then follow up the samples. And when everything is fine, for example, the data QC result, pre-processing QC results are, for example, satisfied, then data can be processed and the results can send to the bioinformatician for analysis, or you can do your own analysis yourself and update the status. So we created this kind of system now to track metadata together with all the process data information. So what it is doing, how we are doing this, so we created this kind of system. In the future, you can think in your own projects, if you have a special large project coming from different people, different organizations, different geographical locations, what you can think is you can define people. For example, you collect data from a different part of US, for example. They enter the data and send you all the data to UMass. And then when they're entered here, you got the information, and then you started doing your libraries, prepared them, and then send the sequencing facility, they sequence. So every single step will be updated here. And then when the sequence come back from sequencing facility, so when you enter those information to Dolphin Meta, it sends all that information to DolphinNext and process the data there and send the results back. And then The portal is another application actually showing the results to your community. For example, you are working with a disease, like we have, for example, skin disease, skin portal that the people in our group are working on vitiligo patients. So what they are doing, they enter all this information to the meta DolphinNext process single cell data and sends this data back with process data, and they go back to the portal, and the community can actually visualize the results and download some of the samples here, some of the patient data, and do some other downstream analysis themselves. So this is a kind of way we are envisioning how it's going to work. So we are trying to integrate data processing with metadata tracking and portals. So there is, if you're curious about these things, we can actually give you another demo about this portion. But basically, some people are entering this data using these Excel sheets to the system, or they can use the forms here. We have some event forms to update status of the sample, and whatever it is here is accessible with its own community. So it means, for example, if you have one community that access only vitiligo patients, they can only access the vitiligo, but there are lupus community, they can access only lupus data in the same portal. But if some communities actually belong to both communities, they can access both data. So we have like a different licensing and different permissions in the same projects. So, but how we are doing this, from collecting sample, sequencing, data processing, and analysis in this whole cycle, actually, we created all the system in FAIR standards, like it's called findable, accessible, interoperable, and reusable standards. So when you are looking for a system, data processing system, to large with a community, to share your results with a community, you have to check if they support FAIR standards, because this means when you need to access to any data in your database, it has to be secure and open to the community that when you have the access. So you cannot access to everybody. You cannot give the access to everybody. So yeah, these has to be, or they have to also be findable. So it shouldn't be like slow, or it shouldn't be like something that, okay, they are there, but there is no way to access the data. You cannot find them in the databases. So we created like web APIs. Those are like called secure token-based web APIs, like Google is using in their own security authentication authorization. We have the similar things here. And also, by creating these kind of standards in ontology-based metadata tracking system, means when you have dictionaries while defining your samples, for example, RNA-seq, just that word can be written with many different formats. So one format could be like, RNA capital dash S is lowercase SEQ, for example. When you define this RNA-seq like this is one thing. But if you don't use the dash, it's still RNA-seq, but the machine cannot understand they are the same thing or not. So machines are not that intelligent yet. You can actually write some code to understand, but it's a dangerous process that to think that RNA-seq without dash and RNA-seq, in that case, they might look like similar, but in other cases, they might not be. So here, what ontology-based metadata tracking system is just forcing the user to use the same wording for these kind of definitions. And in the future, when you do any analysis on them, for example, when you choose only RNA-seq data coming from lupus patients, then all the RNA-seq data is going to be there for you. Not for, like, for example, but those RNA-seq data will be like RNA-seq. You can't see RNA-seq without dash in those RNA-seq data. So they'll be collected. But when you don't have this kind of system, the problem is usually other people are working a lot just to fix these kind of problems in their system. So we are trying to make things more efficient here. And also we created more interoperable and reusable metadata that the people can access, and well maintained, and describe what they are with the descriptions, And when the people goes there, they can actually understand and reuse the data. This is the actual goal. And the integration between different projects, like the example I gave you, like lupus and vitiligo, even they are different communities, they are maybe different patients, but some communities, if it's because they're autoimmune disease, some people might want to integrate both of those results to do other operations on that data. Or there could be like proteomics data and transcriptomics data that you might want to integrate them. So when you create these kind of metadata tracking system, you can actually have a possibility to integrate different type of data in your system by using similar or same annotations, same gene names, or map gene names in with the protein names, or those kind of things will allow you to integrate different type of data. Okay, so DolphinNext it has these functionalities: reusability, reproducibility, and shareability that we when we process the data. So all the steps that you see here are reusable. So when you have, Onur is gonna do the demo. So when you have this step, when you drag and drop, you can actually reuse all steps. You don't need to reinvent the wheel here. Star aligner will be there for you with all the functionalities, all the parameters. You don't need to write the command again. Just you need to put this in and connect with your own pipeline and reuse. But sometimes when you have the star aligner, you have other things like summary and some merge BAM files. If you split this BAM files to increase the speed of your alignment, and then it merges that. You don't need to know how you merge, how you align too much about here. You want to align with star with these parameters. You want to know that. But in detail, you can still go back and see the source. But what you need to do, once you understand what we are doing, you don't need to rewrite the same thing again or execute the same command again and again on the cluster by yourself. You can just use this module, like, we created this whole thing as a module. When you put the module, it supports all these steps in here, and it just simplifies how you look all this pipeline, because when the pipeline becomes complicated, to understand what's going on there becomes extremely hard. We are just trying to make it more modular and group them, that when you look at the pipeline, you will know what you have here. And reproducibility is doing by versioning system here. So we keep all the revisions you have while you're running your pipelines on DolphinNext. Means here, when you run your one of the processing with revision 0, for example, they're going to stay there forever. So when you come here back 2 years later, when you reprocess your data, it's going to be exactly the same processing with exactly the same versions of the software in your pipeline. So this will give you go back and forth to process your data. If you want to reprocess your data with the new revision, you can always change the revision in the pipeline and run again by clicking this button, run button here. So here, what you can do, like when you want to, there's also copy, duplicate, run buttons here. When you duplicate the run, it is going to actually duplicate all the run with all the parameters with that similar version, or it will ask you which revision you want to use. You want to run exactly the same pipeline with exactly the same revision, or with the new revision. So you can actually choose while we are rerunning your pipeline. And this will give you flexibility. Sometimes what they are doing, for example, it happened a week ago that one of our collaborators, actually, he ran everything 3 years ago with Top Hat. And what he wanted to do, he wanted to run everything with star. So it's the same pipeline. We just change the revision, change the Top Hat to star, and then get the results without doing anything. The files were already there in the cluster. So, and we upgraded the annotation files that he used. And in 3 years, he ran lots of samples actually, but with just a single pipeline that we ran all these previous files with the new pipeline, and then they become standard. And then he finished his analysis again, and then published his data like that with the new annotations, with the new pipeline. So this will help you a lot in the future. Once you analyze your data, you just don't want to compare like different pipeline results with different like generated with different programs, because sometimes you get the results online and they give you only TPM files, for example, or some row counts. You want to compare their data with your data, but you don't know how they analyze the data. What we do usually, we just get the raw files, reprocess data, and then compare the same type of data, and then do the batch effect correction like that. Otherwise, you're introducing a lot of bias into your comparison, that even you find some meaningful results, maybe they are not meaningful. And when they send me papers like this, we got this results and then analyze data. This is one of the rejection point for me, that when I review papers, I don't accept that. So they have, unless they use exactly the same methods and parameters while they're analyzing their data, then I accept. Otherwise, how you analyze and how they analyze even the same type of data might not be the same. And again, when you copy, when you're copying the run, it says like keep existing revision or use new revision. So you have this. And shareability, you can share all the pipeline that you created or the pipelines we created on GitHub. And on GitHub, all the pipeline will look like this. The people can actually see, and it's testing automatically. If you add the necessary files there, the people will know that your pipeline is still passing all the tests and in usable format, and they can actually use. And the permissions, you can share your pipeline with everyone, or you can choose only yourself or only your group. So you can create multiple groups and share your pipeline with the people, any group you want here. And we also have this thing here. When you select this publish yes, and then you can define a date. So on that date, actually, it's going to be available to everyone. So you can actually share the link of this pipeline in your publication. The people actually can click and then see what kind of run you did here with the whole run parameters, what code you ran, and with which versions of the software and everything. So you can actually see the program versions in the pipeline, and the people can reproduce your results. And they can also execute their own files in their own environment, but by defining their own clusters in our interface. So for example, in our interface, I defined our cluster, a local machine, a Biocore machine, and Amazon. So I can run exactly the same pipeline on any of those. So when our cluster is busy, I run them on Amazon, for example. And you can monitor the results in your system, in your group, in this run status screen. And when you click, you can go to the run, or when you click the pipeline link, you can go to that specific pipeline, and you see the status here. If it is error, you click the run name of your run, and then go to that run, go to log pages, and see the problem and solve. If you cannot solve, send us an email, we solve, or we are always watching them. If there's a problem, we solve. And also easy reporting. We have the reporting section that you can do the QC. You can see the QC results or alignment summaries. If there's something wrong, you can easily detect here. And they finish extremely fast. And you see the results. Usually if it's a normal, like a standard or an ASIC, you can see the results less than 4 hours in our cluster. So this was an example that Hang in Wallout Lab, this was an amazing case I liked, because he was a bench biologist and he created his own pipeline and reutilized some of the modules we created. And he created his own modules by himself, like all of these things here on the right, his additions to his pipeline. But these star module or BAM analysis module or adaptive trimmer modules were ours. So he didn't need to change them. He just used them and then added some like downstream analysis or processing here. And he tried this a lot, like, years, actually in 2019 to 20, he improved this pipeline. And in the last version, actually, they published this on a paper. So he did a great job by himself. And I really appreciate these kind of users. And another, like Pranita actually was working with us, and she also utilized our platform a lot. And for example, she was mapping, aligning everything with MM9. She needed to upgrade the build, mouse build, genome build to MM10, and she did it very fast. And she did the same thing for HE19 to 38. So because we support all of them. And also she had it like 300 plus RNA-seq data sets, and she ran everything with the same parameters with the updated pipelines in a couple of days. And I also helped here in this Fitzgerald project that they had link RNA quantification. So they used an external resources to do this initial analysis, and they updated their link RNA set, and they wanted to do exactly the same analysis again. They just sent me the link RNAs. And I spent a couple hours, actually 1 or 2 hours, to add those RNAs and then quantify and give the results back, and saved a lot of time and resources, I guess. And also, I did the same thing for another project that they had like 170 patients for vitiligo project, that they wanted to realign everything with the new parameters. And I did everything in 1 weekend. Actually, I ran 160 single cell RNA-seq data and finished all the runs. Actually, it was going to be 1 day, but there was one error in one file, and I need to spend 1 more day. So in the weekend, I finished everything. So it helped me a lot. And also, when you train people and share the results with other people, actually, this platform will help you a lot. And for example, we had this HiC pipeline. They use a similar things for Sprite. So there's similar pipelines that actually you can utilize when the pipeline is adaptable to others. So you can easily do those things. So now it's the demo